Well, hello. Thank you very much, LA, for coming on to give the good people in the community OMF update of the kind of the research that you are doing at the moment for MECFS. Thanks for being here. And I remember the first time that I actually saw you talk about the research, and that was in this Montreal symposium that you, I think, had a big part in organizing back in 2018. And I think one of the most interesting things about what I at least stuck out in my mind was the fact that you were utilizing this technology to create a mild stress response in the MECFS patients that you were measuring to look, or really to characterize post-exertional malaise. And I thought what was really clever about that is that you created this standardized way of creating the stressor and using that as a comparison rather than getting each individual patient to stress of themselves in a way that might be differential. And I think in that capacity, what you've created there may be a useful way to for many other researchers to kind of look at post-exertional malaise, if they could use it in a consistent way, and even to kind of work maybe even diagnostically or in any way of monitoring patients or monitoring post-exertional malaise in patients for all different types of studies or clinical trials. So I thought that was super fascinating. And I thought maybe you could start there and kind of describe that device and some of the research you did around that time.
Hi, Chris. First of all, thank you for the opportunity to be interviewed by you. So yes, we develop a stress test to reproduce or induce in a very standardized way post-exertional malaise. First, when I embarked myself in my research team in this new field for me, I listened to patients, and patients really insisted to the most important aspect of the disease, the post-exertional malaise. So whatever I intend to develop at that time must be centered around the post-exertional malaise. So that's why we reuse a technology that we tested in the past in the lab by using this mechanical stimulation using an inflatable cuff that you apply on any body part, but in our case we use the arm because it's more useful. And we developed this stress test, which seems to mimic a kind of exercise response to the patients that leads to the induction of post-exertional malaise. So first, after over 200 participants that we tested so far, we clearly demonstrated that yes, we induce post-exertional malaise. Yes, the patients are a bit more sicker for a few days, but we are doing in a way that relatively safe. So we didn't have to call an ambulance or to hospitalize any individual following this test. So that was one of the priority number ones, to be able to do it in a safe way. But as you know, if you induce post-exertional malaise in an MECFS patient, they will be a bit sicker. So we reach, I think, safely that point. Secondly, by the application of this stress test, we hope in a single test to reveal what exactly is happening in a patient. So, and as you know, the patients are coming from different type. So clinically speaking, they are very heterogeneous. The trigger factors that led them to MECFS is different in some cases. We know that the vast majority develop MECFS following by a viral infection, but even though the virus can be different, and there are other causes that led to MECFS. So that's why we need to figure out something that can be able to, independently of the trigger, independently of the illness duration, independently of the medication taken by each individual, we need to perform a pragmatic test that can be able to decipher the response and lead us to the real molecular signature that is associated with MECFS. So we developed this test, and each individual become his own control because we have, after 90 minutes stimulation, we draw blood, and before a stimulation, so we have blood, so we can compare changes between each individual. So which is one important point. The second important point for us at that time was to be able to reach out the most severely affected individual, which are the individual that are rarely being tested because they cannot access to the clinic. They are incapacitated. So 85 to 90% of our participants are housebound. So which means that we are sending the clinical nurses at home to do the testing. And that's why all the apparatus we are using are portable, and they are relatively cheap. So it's not very expensive a machine. So which allowed us to do the test. And of course, we are measuring different factors. So we start, as you know, by measuring circulating microRNA. They are very small molecules that are non-coding, but they are very powerful regulator of many genes. So normally you might expect that one microRNA could regulate up to 200 different genes, and some gene can be regulated by more than one single microRNA. So very powerful regulator. They are very well studied in molecular medicine as a biomarker for different diseases, especially for complex diseases, and I think ME/CFS is a complex diseases. So to make a story short, and following our publication in scientific reports, I think we were able to stratify patients in 4 subgroups that are associated with some symptom and symptom severities. So right now the test is accurate up to 94% to predict blindly who is suffering of ME/CFS versus being a healthy control. And what we can do also is the test can also predict the therapeutic response. And I think this is the future of the test, not only to be able to identify symptomatic individual with a precise and rapid diagnosis, but also to try to establish a kind of prognostic. Should I put this individual, for instance, on Amplygene, or should I select something else? So this, I think, can make a kind of revolutions that are wait to be replicated by others, but this is where we are going in the next phase of this development.
Yeah, I mean, there's a lot of information there. It's a fantastic study, and I do recall, yeah, that publication you had in Scientific Reports, which is a fantastic journal. I think that the interesting part about some of this work was the microRNAs, and I'm not sure whether everyone is familiar about exactly what they are. I think you went into a little bit of a brief description, but I know from myself, and I kind of think I follow a similar logic in terms of the research with yourself, in terms of looking at symptoms and metabolites, which metabolites are kind of an expression of genes, but also an interaction with the environment. And I believe that microRNAs probably fit a very similar bill to that. It's just a different area, but they're both very closely associated to the phenotype of a person, which makes sense when you're characterizing symptoms. So I thought maybe, yeah, we could touch a little bit more on this microRNA work. I think that if there's any detail, I guess maybe describe how the microRNAs are generated, and potentially what they could be, also what the impact of them is on the host.
Yeah, so as you may know, 98% of our genome was so-called junk, okay, a few years ago, okay, maybe a decade ago. And right now we know that only 2% of the genome code for something, okay, for gene that would produce a protein or a peptide. And we and others realized that this so-called junk code also for many non-coding RNA. So we have 2 classes, the long one, which are about 200 nucleotides in length, and you have also the smaller one called microRNA, which is about 22 to 26 nucleotides. And the way it works, and I don't want to go in the detail of the biogenesis of both because it's way too complex, but the points to understand is, especially the small microRNA, they are attaching to the messenger RNA, which will be eventually translated in the what we call the ribosomal machinery that will read the copy from the DNA that we call it messenger RNA that will lead to a protein. And by attaching at the end of the messenger RNA, that blocks the decoding. So the machine, the photocopier is blocked. Okay, so in a non-technical term. And also simply the fact to attach to the messenger RNA, the messenger RNA don't like to this type of duplex, and often that brings some signals to degradation. So sometimes you see the messenger RNA start to be degraded, there will be no protein, or we jam the photocopier, if you wish, and the ribosome cannot decode the protein. So such attachment, and you can have many sides, so that can really block completely the translation, and you will see a dramatic decrease in the protein that should produce. So if it's an important protein, you will have a dramatic effect that can lead to more fatigue, to brain fog, to orthostatic intolerance. So altogether, they may be the cause of the, let's say, hallmark symptoms of ME/CFS.
Yeah, I mean this is kind of the interesting research that I guess one of the more interesting aspects of what you found was actually those subgroups, and I think that's kind of important because you're thinking about the disease in different ways. It could be produced or in different capacities or in different kind of packets, I guess, for the different individuals involved with ME/CFS. And so I think if you can kind of subgroup them, that might be a better way to kind of understand their biology. I think because the diagnosis is based on symptoms, there's no guarantee that the underlying biology is going to be exactly the same. And so we have to kind of think
I
in a
agree.
way
I
that's
agree
not.
that with the way. Yeah, sorry. I agree with the way that you summarize this. So we and the clinicians in particular already noticed that we are facing to a very clinically heterogeneous disease. So technically, ME/CFS, even if we regroup all those symptoms, is not truly a single disease or single entity. So there's a kind of spectrum of the symptom, and that makes a lot of sense because there are different triggers that being involved that leads to the phenotype or the disease. So now having more objective molecular markers that can sustain this view and having this group, that will allow us to have a more profound understanding of the pathophysiology of the disease, but also to better select maybe some therapeutic options, so and prevent to use something that it's quite unlikely that the patient will respond because having an elevation of this or a group of macroRNA, the target is disappearing. So how I can connect a drug on a target if the target is not there? As the opposite, maybe some microRNA by decreasing the target pop up, and now you can now use a drugs on this very specific group. So I think if we understand the capacity, having a good or better knowledge of those markers, open up more therapeutic options using existing drugs. So I'm not even thinking about developing new drugs, but using existing drugs. So that can have a tremendous impact in the way that researchers and clinicians will, in the near future, design clinical trial. It's like now in oncology. You know, in oncology, you cannot launch a drug anymore if you don't have a theranostic. So a test that will predict who will respond to your drug and who wouldn't. So the FDA and other regulatory agency won't authorize your clinical trial or the development of your new drug if you cannot select the patient. So I think what we are doing and other colleagues are doing is to bring ME/CFS in the era of precision medicine and to apply this, and that I think that will increase the success of the next clinical trials, and I think it's very important.
And so I guess one of those subsets or one of those subgroups that you found was quite, I mean, a number of those have been quite interesting. Did you want to unpack any one in particular to kind of go into more detail about so maybe some of these treatments or some of the aspects of that may be
Yeah,
relevant?
just to sum up about, I will give you an example of what we refer to cluster tree. So cluster tree regroup mostly ME/CFS patients suffering of orthostatic intolerance, often leading to tachycardia to compensate the hypotensions. And by now having this stratification of patient, we look again about different biomarkers that we measured before, like different biochemical marker like thrombospondin 1. And thrombospondin is of interest for us because it's a powerful vasoconstrictor. So in cluster 3 at baseline, you have a high level of thrombospondin, which means that your arteries are constricted, which reduce the brain blood flow. And after the post-exertional malaise induced by our stress test, there is a drop in the thrombospondin level, often around two-folds of the initial 90 minutes before. And that is sudden to open up the artery because now you create a vasodilatation, and this surge of the blood flow will create a hypotension. And this is when often the heart will react. So the pump will activate to compensate, and you won't like it because that creates a tachycardia. So knowing that, we can now pinpoint this group of patients in cluster 3, as opposed, for instance, patients in cluster 4, who they are going in the opposite direction. They start with much lower quantity in their blood of thrombospondin, but after the post-exertional malaise, the thrombospondin increase, and now vasoconstrict, and now this is where brain fog is creating. I reduce your brain blood flow. So very easy to explain, very easy to demonstrate by measuring those physiological change using the smart biometric vest like Exoskin. And also, we saw the change in the brain oximetry with using another technology that confirmed that we saw the decline in some patients on the oxygen level that suggests that we reduce the brain blood flow as part of the stress test that is inducing post-exertional malaise. Now, what I'm doing with this information is that there are ways to regulate the level of thrombospondin. There are ways to block the receptor where thrombospondin is acting, and there are non-pharmacological and pharmacological treatment for that. So that's why I think that will be the next logical phase for us to launch specific clinical trial on selected individual using our tests to select them, and now to prove whether or not this is efficacious or not. So we need to do a randomized clinical trial to do it, but the big difference is we will select not the whole bunch of ME/CFS patients because you will fail. This is a very select and specific subgroup. So I think we will increase the likelihood of success doing that. And I think this is why I think it's very important.
Yeah, I mean, I agree 100% that. I mean, that strategy that you have taken, I think, is extremely clever as a way of kind of getting around some of the complications we've faced. I mean, there has been a few clinical trials on ME/CFS for different drugs, or one very famous one. And I guess you can tell that there are, benefits for some and not for others. And then this is a way of kind of adding some science to that, having that prediction, and then going into the trial, you can involve that prediction model that you have, or using your tests to identify those patients who will then very well more likely respond to this drug. I think that's a fantastic way of doing it, because then you're going to get people involved. The FDA can come through and approve and all that sort of stuff. So if you can go through, I mean, that process over and over, that would be great. And so it sounds like you're starting with the clusters in regards to thrombospondin 1 and relating that to certain type of treatments. I know another project that you've been working on is in regards to DNA methylation and its involvement in ME/CFS. Did you want to potentially introduce that project and maybe describe some small aspects of that?
Yeah, this is a sub-project in our research program called Maestro and Me, where we are looking at all the multi-omic aspect of the disease. And beside the microRNA, there are also other epigenetics effector called the DNA methylome or DNA methylation changes. So, and we decide to get more insight about those changes by studying several couple of identical twins, but discordant for ME/CFS, which means that one twin is affected and the co-twin is not affected. This is a very, very powerful strategy to set aside what is the genetics component of the disease and what is the epigenetics, so which is coming from the environment. So as you know, the genome of identical twin is they share about 98% of their genome. So we could easily exclude that if the disease is in one twin, this not is due to genetics, but mainly to epigenetic changes. So this is one thing. So, and thanks to Open Medicine Foundation and other association of patients, we were able to collect over 15 couple of discordant twins around the world. So, and because they are localized everywhere in the world, we cannot draw blood easily and send nurses or try to locally organize a clinical setting to receive them. So we use the collection of the saliva. So the saliva is a minimally invasive collection. Everyone is able to spit in a tube, seal the tube, and send the envelope back to Montreal. And very, very stable when the saliva is in the tubes, even at room temperature, so which is one of the advantage. And the second thing that beside the pragmatist, again, the using of saliva, the methylome from the epithelial cell present in the saliva is mimicking more closely what is happening in the brain and the muscle than what you can see using blood cells. So I'm not saying blood cells are not important. In fact, blood cells are very important. We know immunity is involved, but you have a stronger heterogeneity. You have different level of different blood cells. So when you combine all of them, that might change quickly. And others before me, I'm thinking about Patrick McGowan in Toronto, studied very well DNA methylation using PBMCs, or others in Australia, your colleagues that study NK cells methylation more specifically. So because methylation patterns are specific for a cell type or a tissue, so we have to be aware about that. So I think we are the first using saliva in the context of ME/CFS, of course, and because it mimics or seems to reflect what is happening in the brain and muscles, so two important tissues for ME. So we did again a gamble to continue to use that, and we use the twins as a discovery cohort, and now we are in the process to validate